Okay. Hello, everyone. Thank you for coming to our presentation. My name is Elizabeth Liu, and I'm a third-year biochemistry major.
And my name is Anthony Wong, and I'm a fourth-year biology major. So we have been working under the guidance of Dr. Garrett Anderson, and today we'll be showcasing the work that we have done. Our presentation is titled Developmental Electrophilia II Mediated Control of Neuronal Morphology within the Entorhinal Cortex Hippocampal Circuit.
To start off, these are the key points we will be introducing. To best explain our research, we will break our presentation down into two components: Electrophilia II protein localization and spine density quantification. For each, we'll provide some background information as well as the methodology and obtained results.
So we're primarily interested in the cells that comprise the brain, also known as neurons. So neurons are a part of a diverse family of cells. They are assignable based on their various morphological characteristics, and they can be classified as either excitatory or inhibitory. A common excitatory neuron type in the cortical and hippocampal brain regions are known as pyramidal cells. Pyramidal cells have a pyramid shape of cell body, hence the name, and they're also the most common type of neuron found in the cerebral cortex. Pyramidal neurons are particularly spinous, and they have many branches, and this also lends well to the role of pyramidal neurons as sites of information transmission. And on the right, here's a diagram showing how neuronal communication works. So we have the axon, which is where information travels down the neuron. Information flows out of the axon from the presynaptic vesicle. The presynaptic vesicle contains neurotransmitters, and these are chemical substances that are used to transmit information to other neurons. When the neurotransmitters are released from this presynaptic vesicle, they travel to other neurons and they are received by receptors on spines. And this allows information to be communicated to other neurons. So to further elaborate on the structure of pyramidal cells contain three distinct compartments. There is a primary apical dendrite, there are secondary apical dendrites, and there are also basal dendrites. The primary apical dendrite emerges from the apex of the cell. The secondary apical dendrites emerge from the primary apical dendrite, and basal dendrites emerge from the base of the soma. Each compartment also has functional differences.
There are many proteins and other biological molecules that affect neuronal structure and function. One such protein is electrophilin. So electrophilins are a family of cell adhesion G protein coupled receptors. Cell adhesion molecules mediate interactions between neurons by binding with other molecules, which leads to a series of reactions which ultimately can change neuronal structure and contacts. An oversimplified way of thinking about cell adhesion molecules is they could be seen as anchors, which help facilitate the connections between neurons. Without them, the connections between neurons may be incomplete or disrupted, which is why many neuroscientists are interested in investigating the different families of adhesion molecules. So electrophilins were initially identified as receptors for alpha-latrotoxin, which is a toxin found within black widow venom. Since then, electrophilins have been identified as a molecular contributor to the formation of neuronal networks and influence synapse formation. Their function as mediators of adhesion events provide insight into their contribution to development of synaptic connections. Consequently, defects in the electrophilin gene have been associated with ADHD, autism spectrum disorder, schizophrenia, epilepsy, and other neuronal disorders. Specifically, our research focuses on electrophilia II, which is an isoform found in mammals. Anderson et al. Has shown that the number of excitatory synapses in the hippocampal region are reduced following the genetic deletion of electrophilia II, suggesting that the lack of electrophilia II causes changes in a neuron's ability for target recognition. Specifically, Anderson et al. Focused on the SLM region of the hippocampus and found a decrease in spine density. To see if this ability was reflected in other regions, we decided to look at the medial entorhinal cortex, also known as MEC.
So within the brain, there are many circuits that are connecting different regions of the brain and which give rise to specific neurological functions. Our research is centered around the hippocampal entorhinal cortex circuit. So the hippocampus is a brain structure that plays an important role in learning and memory. And the entorhinal cortex is an area of the brain which acts as a network hub for information related to memory, navigation, and time perception. and the MEC is a region of the brain that is highly enriched in lateral filament 2. So Santiago Ramon Cajal, who was the first neuroscientist, was interested. He discovered the circuit shown here, and he illustrated how complex the connections between the two structures are. As can be seen in the circuit, the entorhinal cortex, specifically the medial portion, plays a major role of being the main input into the hippocampus, and neurons in the layer 3 of the entorhinal cortex influence the hippocampal neurons. In particular, in the circuit, we are focusing on the connections between the entorhinal cortex and CA1.
Previous research has shown synaptic connections to be highly specific, but details about the molecular mechanism and contributors require more investigation. Here, we will be presenting on the role Electrophilia II protein plays in controlling cell type and regional specific patterning in definable MEC pyramidal neurons.
So we quantified where electrophilia was localized, and we found that electrophilia 2 is enriched in the dorsal hippocampal region of the brain, a region which is associated with spatial learning. And to quantify this, we took measurements of electrophilia 2 signal intensity across hippocampal regions, such as the dentate gyrus, CA1, and the MEC using ImageJ. We normalized the intensity by dividing all the values in a given layer/region by the highest value from that brain. We performed this analysis on brains from ages P0, P7, P14, P21, P30, and P60 to determine how the localization of electrophilia 2 changes during development. And so we found that the electrophilia 2 signal was brightest in the dorsal region as compared to the ventral region, and that the MEC gradient is established before the CA1 gradient. So looking from the dorsal to ventral MEC, we see that in the SLM, it is enriched in that electrophilia 2 enrichment coincides with the timing/development of the SLM. And we see that the brightest signal is in the SLM in the most dorsal portions, and it gets dimmer in the ventral portions in the MEC. And we also see that the MEC gradient is established before the CA1 gradient. So looking at figure A, so for the white boxes in the figure, these boxes denote the SLM area of the CA1 region and also the MEC. And this is shown throughout the developmental time span. So we see that electrophilia 2 enrichment changes throughout the development in each time point. And as you can see in these graphs, in the CA1 region, we see an increase of electrophilia 2 enrichment across the developmental time span, while in the MEC, the levels of electrophilia 2 enrichment are consistent across development. So as a result of our protein localization, we now know where electrophilia 2 is located, as we found that electrophilia 2 is enriched in the SLM and MEC. So from there, since we established that electrophilia 2 coincides with the timing of development, we wanted to see how development would change if we were to remove electrophilia 2 in the circuit. And this also leads into the next part of our research, in which we knocked out electrophilia 2 in the MEC.
Now that we located where electrophilia 2 was expressed, we were able to determine the coordinates needed for targeted deletion. We utilized the genetically engineered electrophilia 2 conditional knockout mouse model. In combination with stereotactic injection of pre-recombinase expressing adeno-associated virus, we achieved targeted MEC-specific deletion of electrophilia 2. In this figure, the pre-recombinase virus was visualized with TD tomato, which fluoresces red, while electrophilia 2 was labeled with green. The virus is localized to the MEC, shown in panel A, and where the virus is present, electrophilia 2 is no longer present. Within the MEC, a gradient of the pre-recombinase and electrophilia localization was observed. In particular, layer 3 of the MEC showed the greatest decrease in electrophilia 2 signal intensity. Overall, only the superficial layers of the MEC were affected, while the deep layers of the MEC showed very little change in electrophilia 2 expression. With electrophilia 2 expression levels showing the greatest changes in layer 3, we wanted to determine whether removing electrophilia 2 in this region would lead to any changes in the formation or maintenance of synaptic connections. Synaptic connections between neurons are facilitated by dendritic spines, and morphological analysis can be used to determine if changes have occurred in a given circuit. So the study of morphological structures in neuroscience is a relatively new area of study, spanning a little more than 100 years since the first time a neuron was stained. Despite the identification of many key functions, there are still aspects of the neuron that remain less understood. One such aspect are the dendritic spines. So spines are projections from dendrites, and they're crucial in receiving and processing information. Their most interesting function is serving as the main site of excitatory synaptic transmission, which is why analyzing spines has been a key method in determining neural circuit remodeling. Knowing this, determining whether or not spine density would change if latrofilin 2 was removed was an important step in determining whether latrofilin 2 was involved in synaptic development in the MEC.
And so to answer this question, we injected the Cre recombinase virus into the genetically engineered latrofilin 2 conditional knockout mice. And we utilized two groups, one knockout and one control, in order to determine if there were any differences. And after retrieving and sectioning the brains, neurons were imaged using confocal microscopy. And in particular, we focused on the deep and superficial regions of layer 3 of the MEC, with a primary focus on the pyramidal neurons. These confocal images were processed and reconstructed in Imaris, which is a 3D reconstruction software.
We discovered that there were significant differences in spine densities in the deep and superficial regions of layer 3 pyramidal neurons of the MEC. For superficial layer 3, we saw a marked increase in the spine density of the primary apical dendrite. On the other hand, for deep layer 3, we also saw an increase in the spine density of the primary apical dendrite, but we also saw a decrease in spine density for the secondary apical dendrites. We also looked at layer 5, but found no significant differences. From these results, this suggests latrofilin 2 has a layer-specific role in controlling the synaptic connections of the MEC. The increase in spine density at the primary apical dendrite for both the deep and superficial regions of layer 3 is particularly interesting. Taking a closer look at the spines, we see an increase in Philopodia spines. Philopodia spines are immature and transient structures within circuits and are thought to result from testing new but weak connections. Knowing this, removing latrofilin 2 is causing an increase in Philopodia spines, and thus may be affecting the ability of spines to form strong, stable connections. This increase in spine density may be an attempt to compensate for the decrease in synaptic strength.
So in summary, some overall takeaways from our results are that we determined that latrofilin 2 enrichment coincides with the timing of development of the SLN. And as a result of these findings, we wanted to determine how development would change if latrofilin 2 were removed. And when latrofilin 2 was removed, changes in synaptic density were observed in deep and superficial layer regions of layer 3, the MEC.
In general, there are 4 main morphological classes of spines: mushroom, thin, stubby, and Philopodia. Throughout neuronal development, spines can change, they can form, they can disappear, and this ability to change is the basis of synaptic plasticity. Since we saw changes in the overall spine density between control and knockout groups, we wanted to see if there were any differences between the spine classes. Different spine classes are known to have different strengths and contributions to the overall circuit. By determining the densities of each spine class in the presence or absence of latrofilin 2, we can determine if latrofilin 2 also has the ability to influence the strength of the synaptic connections in the hippocampal and terminal circuit. In order to determine this, we looked at layer 2 and layer 3 deep and superficial pyramidal neurons. Following the same technique for neuron reconstruction, we're taking it a step further by using Imaris's spine classification function to label spines based on their morphological characteristics. The morphological characteristics that we are using to classify these spines focus around the dimensions of the head, neck, and length of each spine. Ultimately, every spine was categorized into one of the 4 spine classes: mushroom, thin, stubby, and Philopodia. With each spine class, we'll determine the density by dividing each spine class number by the overall length of the dendritic shafts. We would like to take this opportunity to thank all of the wonderful people who have helped us along the way. We would like to especially thank our PI, Dr. Anderson, and our graduate student mentor, Jordan Donahue, for all of their continued support and guidance. We would also like to thank our current and former lab colleagues for all of their hard work and input. And lastly, we would like to thank our friends, family, UCR, and the Whitehall Foundation for their support as well. And that concludes our presentation. Thank you all for coming.
Great job. Thank you. Again, let's give them a round of applause.